Hi everybody, welcome to this virtual session of the Winemakers Research Exchange. I'm Joy Ting, the Research Enologist and Extension Coordinator for the WRE. If you're new to the WRE, we are a nonprofit organization of winemakers funded by the Virginia Wine Board with the mission to promote innovation through experimentation and education in the wine industry. The main way that we do this is through practical production scale experiments conducted in wineries around the state of Virginia. Experiments are evaluated both chemically and at sensory sessions where winemakers meet together, taste the experimental wines blind, and then discuss the results. We had planned a full slate of sensory sessions for this season. However, at the time of this recording, on March the 23rd, 2020, the CDC and the Governor of Virginia are asking us to avoid meeting in person in light of the global pandemic of COVID-19. Therefore, all of our sensory sessions are on hold at this time. We look forward to being able to meet together in person again, and I will let you know as soon as I do when we have a new schedule planned. In the meantime, I have chosen some experiments that are more driven by data and ideas than by sensory differences to share with you in this format. I truly miss seeing each one of you, and I will miss the lively discussions that usually ensue during WRE Sensory Sessions. But I sincerely hope that you and your loved ones are remaining healthy during this time. I look forward to a time in the near future when we can all meet together again, but until then, I'm going to try to get some form of content to you roughly once per week, either a video session like this one or a more standard newsletter highlighting completed projects from past years. So the set of experiments that we're going to look at today was really born from the 2018 vintage. And if you think back to 2018, you remember it was very rainy. Because we make wine in Virginia, unfortunately, this is just something we're going to have to deal with once in a while. One of the problems with the 2018 vintage was that most of us saw high levels of volatile acidity in the finished wines. And most likely that was due to spoilage organisms that were coming in on the grapes. So even though we were using plenty of SO2 at crush, we still had this volatile acidity. So as a result of that, we started to look to see if there were additional tools that we could use to combat these spoilage organisms. And that led us to two ideas, using Chitosan at crush and using non-Saccharomyces yeast at crush. So the topics for this session are going to go this way. The rest of this video, I'm going to just talk about generally what kind of microbes we see on grapes and why our 2018 vintage might have meant additional spoilage for us. I've broken this into 3 videos, so that will be the first video. The second video will be looking at Chitosan itself, so I'll give you a brief introduction to Chitosan and how it works, and then we'll look at 2 experiments looking at using Chitosan at crush. The third video will look at using non-Saccharomyces yeast, why those might be bioprotective, and then we'll look at 2 experiments using those for bioprotection. At the end of that video, I'll have a quick wrap-up and ideas for some future experiments as well. I would also say that if we were at a real WRE session, each time we talked about an experiment, you'd be able to sort of pick up that report from the middle of the table and see in more detail some of the things about the methodology. I will post those reports on the website, so if you're interested in more detail about the methodology than what I'll use in these videos, you can go ahead and grab that PDF from the video or from the website. So before we get started, though, I think it's important for us to sort of continue to think carefully about volatile acidity itself. And we did a lot of this last year, but again, if we were at a real WRE session, this is the part where I would tell you to take out a piece of paper and brainstorm with your group all of the different places and ways that we can accumulate VA. So what I'm thinking here is just start in the vineyard and say all the different things you can think about where you could have VA or where you need to be careful about trying to control things so that you don't get volatile acidity. So I'd like, instead of doing that with your group, unfortunately today you're going to have to do that on your own, but go ahead and take out a piece of paper, pause the video, and take a couple of minutes to brainstorm some ideas. Okay, hopefully you paused the video and had some time to just start to think a little bit about all the different places and ways we can get some volatile acidity. I'm going to go over the list that I brainstormed. You might have some things on your list that I don't have on mine. That's great. I might have a couple on my list that you don't have on yours. This is why it's better when we all work together. Okay, so the first thing I thought about was anytime we have rot in the vineyard. Now, particularly, we think about that about sour rot, we think about that with botrytis, but anytime we've got rot in the vineyard, basically we're bringing in a lot more spoilage organisms that can lead to volatile acidity. Some of the pests in the vineyard also cause this. So if we have birds that are pecking at our grapes, that releases juice onto those grapes and gives a nice little spot for our spoilage organisms to grow. Fruit flies and bees can also sometimes poke holes in those grapes. And fruit flies and bees, once they get there, they can sort of pick up spoilage organisms and fly over to another set of grapes and deposit those on the next set of grapes. So not only do they sometimes cause problems with the grapes themselves, but they also can spread those problems to other parts of the vineyard. Split berries during rain also will give us higher levels of spoilage organisms, because as soon as those berries split, we have a nice little nutritious environment for organisms to live on the outside of our grapes. So basically, these are all different ways that spoilage organisms can come in on the grapes and cause volatile acidity in the finished wines. Once we get that fruit into the winery, then some of our winery practices can also increase or decrease depending on how we're managing things. So cold soak itself has lots of different issues. So basically, during cold soak, we can see the amplification of Cloacra apiculata. That's that organism that causes ethyl acetate. We can also see the proliferation of other Saccharomyces yeast that would come in on those grapes, and also Acetobacter, which is a bacteria that basically uses oxygen in the production of volatile acidity. And during cold soak, we just don't have nearly as much CO2 being produced, and so there's just a lot more oxygen around. Once we get the fermentation going, we can have some problems with the yeast, and the yeast can also produce VA. So if we have poor yeast nutrition during fermentation, so if for whatever reason our grapes just didn't have enough nutrients and we're not adding enough nutrients, our yeast can get stressed during fermentation and produce VA. We can also just sort of have a bad match between our yeast strain and whatever's going on in that fermentation. I always think first about temperature there. So if the fermentation gets too hot, the yeast can get stressed. But other ways that those yeast can get stressed would be if we have too high of an acidity for the yeast strain that we're using, or if the alcohol levels just get to be too high for the particular yeast strain that's being utilized. Once we get through regular fermentation, we're not out of the woods yet. There's lots of different creatures that can convert malic acid to lactic acid, and different strains of those creatures can have different production of volatile acidity as well. Even if we're using a good strain of malic acid bacteria, if that just takes a long time, you end up with having your wine kind of languishing without any SO2 protection. Basically, if it's not producing enough CO2 to push out the oxygen, and you don't have SO2 in there to protect it, any organisms in there that can use oxygen to make acetic acid have oxygen available to them. Once we kind of get through malic and we get things sulfured, we still have some accumulation of volatile acidity that can occur, basically due to any time that we have oxygen exposure to our wine. So those bacteria that are going to make the Acetobacter basically are still there. And so if we have any oxygen exposure, they can convert that. They can use that oxygen to make VA. That can happen if the barrels are not topped, if we're not properly monitoring our SO2 levels and they get too low. And then basically anytime we're moving the wine around, we're kind of introducing oxygen as well. And that can give a substrate for our spoilage organisms to work. The other thing is we can take perfectly healthy wine and we can put it into a barrel that houses a colony of microbes that can then start to spoil it. And that brings us sort of to a bigger question. Basically, anytime we have poor cellar hygiene, we can be taking good wine and inoculating it with some of these spoilage organisms. And so cellar hygiene itself is a really important thing to think about here. But in today's set of experiments, we are really going to focus on what's happening with these spoilage organisms that are coming in on the grapes. Okay, so what are the microbes that are coming in from the vineyard into the winery resident on those grapes? There's lots of different studies looking at this, and I will tell you every different study kind of tells you different specifics, but lots of common factors. So there was a particular study out of Latvia that I looked at that found when they used molecular techniques to look at all of the creatures that were found on those grapes. They found 11 different genera of gram-negative bacteria, 11 different genera of gram-positive bacteria, and 9 genera of yeasts in there that included 51 species of bacteria, 52 different species of yeast, and of that population, 62% of the population was yeast. It's important to say at this point that that yeast was not Saccharomyces. None of the yeasts that they found in that particular study were Saccharomyces coming in on those grapes. Now, when we look at a bunch of different bunch of different studies, we do see differences in which species of gram-negative or gram-positive, or how many different species we see, but there is a striking similarity of the overall community of flora that are coming in on the grapes. So it seems like there is a very specific kind of set of organisms that you could find on grapes in any given experiment or any given situation. You might find a subset of that particular larger set, but you almost always find certain ones there, and then they're sort of mix and match of the rest of them. And the differences that you find really are in who's dominant due to a number of different environmental factors. So the climate, the altitude, what variety of grape you're talking about, what the disease pressure in that particular vineyard is, what the vineyard practices in that particular vineyard might be, and the seasonal conditions. So for example, we might find different dominance or different numbers based on, or in the grapes that came in 2018 versus 2019, but they would still sort of belong to this generalized group of organisms that are found on grapes. Now, it's important to remember that grapes are not actually, the surface of grapes are not actually a really great place for microbes to grow. They're covered in a waxy cuticle that essentially is there to repel moisture, and all of our microbes need a certain amount of moisture in order for them to be able to maintain the integrity of their cell membranes. So basically what they do is they grow particularly in the spots where the stomata are. So the grapes themselves, the grape skins, have little pores in them that open and close in order to allow CO2 to come in for photosynthesis and allow oxygen to go out as a byproduct of photosynthesis. If that didn't happen, the oxygen would actually poison the inside of the grapes. Now, when that happens, there's a little bit of seepage of grape juice that goes out onto that outer skin of the grape. And it's really around that little spot that we find this community of organisms that make up the microbes of the grapes. However, when we find instances where the rest of the grape does have moisture, that community of organisms can quickly sort of bloom and take over wherever there's moisture. So that means that if there's fog or dew on those grapes, if we have seasons of high humidity, that actually cause some condensation there, and particularly with rain. So whenever you're taking in those wet grapes, that's when you tend to see, for example, clochera causing ethyl acetate, that nail polish remover smell. That's when we tend to see it because there's just been that opportunity for a bloom on the outside of that grape. So that community of organisms that lives around the stomata, they do tend to increase and sort of change a little bit as they go through ripening. When we look at who's actually present there, there can be anywhere from 100 to 1,000,000 cells per centimeter squared of surface area. But when we think about that, they're almost entirely non-Saccharomyces. In order to find Saccharomyces, oftentimes one berry in 1,000 reports any amount of Saccharomyces at all. So when we think about those microbes that are coming in on the grapes, we need to remember that the inoculum of non-Saccharomyces yeast and bacteria that are coming in off of those grapes are probably, they can be as high as whatever you're inoculating with your commercial yeast. That's especially true in diseased grapes. If you have sound grapes, you have more of a competitive advantage with that inoculant. But if you have diseased grapes, or really sometimes just even really ripe grapes, you could be sort of giving your Saccharomyces less of a competitive advantage along the way. So whether you're inoculating or not, that native flora that's coming in off of the vineyard may have a significant effect at the beginning of fermentation. Some of that effect is negative, so they're sort of in chemical warfare with each other, so they might be inhibiting each other in any number of different ways, including throwing off toxic compounds. They might be depleting the nutrition that's in those grapes so that once your Saccharomyces do take hold, they don't have enough nutrients. They might be producing off odors that would not be things that we'd want to have in our wine. But I should also say that they also have some positive effects, and we're going to look at that more in part 3 of the video. But they can also produce some good odors themselves. They can work on the grape itself to help to release some of the precursors of the aromas that the Saccharomyces are going to be making for us. And they can also help break down some of the structural component of the grape to make that more available for Saccharomyces. And so really when we're thinking about these microbes, they are not all good and they are not all bad. It's basically an issue of managing the good and the bad. Okay, so who are these microbes that we find on our grapes? There's lots of them, but I thought I would just give you a quick snapshot of some of the frequently found guests that are coming in. So the number one most likely thing that is coming in off of your grapes is Clochra apiculata, which is also known as Hanseospora uvarum. If you're not familiar with sort of the nomenclature of microbes, oftentimes they'll have these sort of two different names. That's a vestige of a historic time when we would identify these things by what they looked like under the microscope. These look very different under the microscope based on whether they're in their meiotic form or their mitotic form. When we run the genetics on those, we realize that those are the same organism. And so we'll that just, whether we call it Clochra or Hanseospora has to do with which form it's in at that particular time. Clochra is that organism, as I said before, that produces ethyl acetate, which is the nail polish remover smell. But it also does a number of other things. It produces other sort of killer factors that might be inhibiting the grapes around it. And we also need to remember it can produce a whole lot of acetic acid, 25 times the concentration of what we would see from Saccharomyces. And that actually turns out to be a big inhibiting compound for Saccharomyces itself. So if you have a lot of Clochra and it's producing acetic acid, that acetic acid itself is inhibitory to Saccharomyces. So it may actually slow down or inhibit your fermentation if Clochra gets hold. The reason that Clochra is so important is that, first of all, it is usually the dominant organism that's coming in off of the grapes. But secondly, it doesn't slow down as much as other organisms when it's cold. And so when you do cold soak, the other organisms are going to be inhibited more than Clochra is. And so you'll see sort of this growth of the Clochra population even when you're in cold soak, and that can give it a competitive advantage, which you just have to be careful about. We don't want to do that. Another organism that comes in is Mechnocauia pulcherrima. We're going to talk more about Mechnocauia in another video, and so I'm going to kind of just leave that one there to say it definitely comes in off the grapes, but we'll talk about its properties in video number 3. Pichia is another one that comes in. Pichia doesn't do so well. It's not like Clochra. It doesn't do so well when it's cold. But if you have other delays in fermentation, so if you're doing an ambient fermentation and it just takes a little bit longer to get going, or if you don't have a good, a good strong inoculant and it takes a little bit of time, Pichia can go to work. And the main thing with Pichia is that it can produce 4-ethylphenol. And we think of 4-ethylphenol with Brettanomyces, but Pichia also can make that particular aroma compound. And so obviously most of us don't like that. Then we can get those. So those are all yeast that would be coming in. We also have a number of bacteria that are coming in. So when you kind of send your samples off and it comes back and it says acetic acid bacteria, that includes Acetobacter, Gluconobacter, Gluconacidobacter. Those are all sort of lumped together as our acetic acid bacteria. These are things that require oxygen, but they can convert ethanol to acetic acid in the presence of oxygen, which can be a big problem for us. These tend to come in particularly when we have compromised grapes. So if the grapes have botrytis or if they have sour rot, we see much higher levels of these guys. And then we also have our lactic acid bacteria. So as I mentioned before, there's lots of different organisms that can convert malic acid to lactic acid. We generally select for Enococcus ini because it does that without producing a lot of the other off odors and off flavors that some of its comrades can produce. But when we look at who's on the grapes, we see lots of lactobacillus, and we also see pediococcus. And this group of lactic acid bacteria can produce acetic acid. They can produce diacetyl in much higher amounts to the point where it becomes a fault. They can produce compounds that come up with sort of a mousiness, biogenic amines. And when we think about biogenic amines, one example would be cadaverine. So that would not be a flavor compound that you would want in your wine. And they can also produce sort of these big carbohydrate molecules that can cause ropiness in the wine. That's Pediococcus in particular. So all of these things are microbes that are coming in on your grapes. So the question is, what do we do about that? Before we talk about what to do about it, though, I did want to show you this one particular study that just sort of clarified a lot of these issues for me. So in this particular study, they just looked at a population survey. So who are the microbes that are coming in on the grapes in this particular situation? And what they found is when they brought in sound grapes, they had about 6 different total sort of species of non-Saccharomyces yeast that they saw coming in. But when they looked at sour rot grapes, you can see pretty much every single species of non-Saccharomyces yeast that they looked for, they had a positive result for. So when we look at our difficult growing years like 2018, where we do have higher levels of compromise in those grapes, not only do we have a higher number of our spoilage organisms, remember that difference between 100 and 1,000,000, a lot of that has to do with what kind of growing season we've had, but we also have a higher diversity of spoilage organisms, meaning that there's just greater genetic diversity in there and more likely that somebody in there is going to be causing spoilage. So the first thing to be said about this is just really underlying the importance of sorting out compromised grapes, both by hand harvesting in the vineyard and on the sorting table, to keep as much of that as possible from getting into your fermentation vat. Because once those guys are all there, then you're just going to be fighting against them for the rest of the life of that wine. So to understand some aspects of managing these, I think it's helpful to look at what happens to these microbes once we get them into the winery, once we crack those grapes apart, and once fermentation starts. So if we look at the graph here, basically what you're seeing in terms of the population level of non-Saccharomyces yeast, Acetobacter, Pediococcus, Lactobacillus, and Enococcus, those are just reflecting the diversity of what's coming in on the grapes at harvest. And again, that non-Saccharomyces yeast portion, that's more Hanseospora or Clocara than it is anything else, but it can contain any of those creatures that we talked about. So in the first 3 to 4 days of fermentation, there's really a very strong selection for Saccharomyces. So again, we haven't talked about Saccharomyces yet because it's very uncommon on the grapes coming in. So either you inoculate with your own Saccharomyces or allowing the fermentation to start very quickly does select for Saccharomyces. And there's a number of reasons for that. As soon as you open up those grapes, you have a really low pH environment, which is inhibitory to most microbes, but it is more inhibitory to everyone else than it is to Saccharomyces. So it allows Saccharomyces to start out competing those other microbes that are around. You also have a really high sugar environment, and as much as we think about that as nutrition for the grapes, it's also osmotic pressure. So what that means is because there's so much sugar in the environment, basically the liquid inside of the microbial cells is trying to sort of, it's drawn out toward that high sugar environment. So they kind of have a hard time maintaining the integrity of their cells in that high sugar environment. Very quickly, fermentation uses up all of the oxygen that's in the environment, produces an anaerobic condition. And many of our spoilage organisms, they may or may not be able to survive without oxygen around, but they certainly aren't able to produce acetic acid when there's no oxygen around. It also produces a lot of ethanol. Ethanol can start to destabilize the cell membranes of most of our organisms. And again, it does that to Saccharomyces as well, but Saccharomyces is less affected by it by everyone else, so it gives them a competitive advantage. The phenolic compounds in a red wine fermentation can be somewhat antimicrobial and sort of add to the stress that those other microbes are feeling. And high temperature. So again, in a red wine fermentation, when we sort of have these higher temperatures, that also stresses the cell membranes. And again, just to highlight that does that for Saccharomyces as well, but Saccharomyces is better adapted to it, so the competitive advantage goes to Saccharomyces, and these other guys, you can see their populations decline pretty quickly once we get there. But notice they're still there, so they haven't completely disappeared. Now, there's lots of things we do in our winemaking that can affect how long those spoilage organisms persist. So how long they're there and how long they're actually making an impact on what's happening around them. So how long they're able to metabolize. So we already kind of talked about cold soak, but thinking about cold soak in this regard, if basically you're inhibiting Saccharomyces from doing fermentation and you're keeping things cold, you still have that low pH environment and that high sugar, but you're no longer in an anaerobic condition. No matter how much you gas that tank, there's still some oxygen around. You don't have any ethanol, and again, you don't have that high temperature. So you're kind of giving this soup a little bit longer to work. Um, Cool fermentations can also sort of not kill them off as quickly. So again, just one less stress factor for them. And it doesn't mean that you can't do cool fermentations. We do those a lot. We just need to remember that if we're doing cool fermentations, that we just don't have that other stress factor for these non-Saccharomyces yeast in particular. Low sulfur. So if we're working, if we're trying to kind of dial back the amount of sulfur dioxide that we're using, we need to remember that SO2 does knock back some of these populations. And so again, it doesn't mean you can't do that, but just remembering that you're doing less to knock this back so you have a higher inoculant of it. So if you sort of stack a bunch of these on top of each other, you might get to a point where you have a problem. And then our own clarification methodology. So for white wine fermentations, if we're doing a really good job of really clarifying that juice, then we're simply not bringing as much particulate matter over, and we need to remember that particulate matter includes some of these microbes. I'm not saying that you can't, increase the turbidity of your fermentations, but again, just realizing if you do that, that you have a little bit more inoculant of these non-Saccharomyces microbes that are going into your fermentation. So given the fact that we have all of these microbes that are coming in on our grapes and getting into our fermentation, what kind of management strategies can we employ in order to try to get a good clean fermentation without sterilizing the grapes, which we can't do? So our primary way of doing that, or at least the baseline way to do that, is with the addition of sulfur dioxide at crush. So just a quick review here, remembering that when we use sulfur dioxide, it's in a water-based environment. So that sulfur dioxide in water is essentially immediately going to take on one of three different forms. There's lots of different molecules of sulfur dioxide in that aqueous environment, and the different proportions of them will be in different forms here. But the only one that's useful against microbes is our molecular SO2. And if we kind of look at this equation, we just need to remember that the more acidic our environment is, the more we're sort of leaning over to the left-hand side there. So we have a higher proportion of molecular SO2. The higher our pH is, the more we lean over to the right-hand side there, and we have a lower proportion of molecular SO2. So when we think about how effective our SO2 is, our SO2 is going to be more effective against microbes in a more acidic environment, and it's going to be less effective in a more basic environment. So just keeping that in mind. So that sulfur dioxide, again, it's only the molecular SO2 form that is useful against microbes. And the reason for that really has to do with the charge on those different molecules. So if you look at the bisulfide or the sulfite forms of SO2, you can see that they're negatively charged. Well, the cell membranes of microbes are also negatively charged. And so we know that like charges are going to repel one another. And so the bisulfite and the sulfite ions are going to be repelled by the cell, whereas our molecular SO2 does not have a charge on it. And when we look at how that molecule is arranged, there's not even really a distribution of charge within the molecule. We call it nonpolar. And that means that it can basically slip through the cell membrane and get into the cell. Once it gets into the cell, then it can go around and inhibit enzymes, it can denature proteins, and it can inactivate thymine, it can bind to amino acids, it can do all kinds of things that wreak havoc inside of that cell. So it's only our molecular SO2 that gets inside the cell, and once it gets inside the cell, it basically can inactivate or even kill that cell depending on what level it's present at. So if we look at what level though is needed to kill that cell, We need between, and that's on this chart would be the biocidal effect. We would need between zero point 0.5 and zero point 0.9 molecular SO2 in order to kill off these microbes. Well, if we look at it at a pH of 3.6, which is actually probably a pretty low pH for what a lot of our red wine fermentations are seeing, we would need between 30 and 55 parts per million of free sulfur in order to start killing off those microbes. When we add SO2 at crush, we need to remember that that SO2 is binding to all kinds of different things. So it's binding to the sugar itself, it's binding to the grape skins. And so it's going to be very difficult to get to a 30 to 55 parts per million free sulfur. The amount of sulfur we would need to add would be very, very large, and it would cause other problems. So when we add lots of SO2, um, it can inactivate thymine, which is a micronutrient that's needed by our non-Saccharomyces or our spoilage organisms, but it's also needed by Saccharomyces. So we don't want to inactivate all of that. Initially what it's going to do is it's going to increase the extraction of color in the finished wines. It's going to bleach color. But if there's a lot of SO2 still around once you start fermentation, that becomes a reducing environment, and you can get these reduced sulfur compounds like H2S, rotten eggs. And if the yeast kind of get a hold, if that affects the yeast too much, what they do is they detoxify it by producing acetaldehyde to bind it up. And so that's just going to mean we're going to be carrying a lot higher amount of total sulfur in the wine. And so really, there's a limit to how much SO2 we want to be adding at crush, regardless of how many microbes we have, because it can cause other problems in our finished wine. The other thing we don't talk about very much is that, if let's say that you've got, a population of however many, um, clockera in there, and you kill off 85% of those. Well, the 15% that remain are the ones that are more tolerant to SO2 than the ones that you just killed off. And so what you're doing is you're just selecting for these SO2 tolerant microbes. With clockera, that becomes not so important as fermentation goes on, but with some of our other spoilage microbes that do survive and can spoil the wine during aging, like Acetobacter or Brettanomyces, we really want to avoid selecting for those SO2 tolerant microbes. So when we look at these vintages like 2018, and we've got all of these microbes coming in on our grapes, what is it that we can do in addition to SO2? And that was really the question that was driving the experiments that we're going to be talking about in the next two videos. So the two options that we started to test were, a lot of us have used Chitosan during aging. Would Chitosan be a tool that can be used at crush? And there are a couple of products that are now being marketed to be able to do that. So we have a couple of experiments looking at that. And then some folks, we talked about non-Saccharomyces yeast as being spoilage organisms, but if you choose which ones in particular and select for certain characteristics, some non-Saccharomyces yeast are being used for bioprotection, particularly during cold soak. And so we have a couple of things testing that as well. And those are what we're going to be covering in videos number 2 and 3.